Hi, I'm Dr. Skander Hathroubi, a postdoc at UC Santa Cruz, and today I will talk about the fascinating world of biofilms. So I will start with a short introduction about biofilm and talk about their importance in clinical, public health, and industry. Then I will talk about biofilm formation and its composition. I will also briefly talk about some methods to study biofilms, and I will give some example of the impact of biofilms mode of growth on bacterial life. Finally, I will end this presentation by talking about some of my current research on Helicobacter pylori biofilm. So what's a biofilm? Everything started in 1978 when Dr. Bill Costerton established a profoundly new microbiological paradigm, the biofilm theory. He published an article in Scientific American in which he stated that bacteria stick on available surfaces in glycocalyx enclosed biofilms. The sessile bacterial population, or biofilm, become predominant in natural, industrial, and particularly in medical ecosystems. So where we can find biofilms? In fact, biofilms are everywhere, around us, among us, and in us. Here are some examples. You can find biofilms in your bathtub, in your toilet, on your teeth. Biofilm are highly associated with implant-associated infection. Biofilm, in fact, can colonize catheters, prosthetic heart valves, and urinary devices. And they are also associated with several chronic infections such as Lyme disease, otitis, and cystic fibrosis. In addition, biofilm are also linked to nosocomial infection, which are hospital-acquired infection. Nosocomial infection are the fourth leading cause of death in the US, with around 2 million cases annually. And about 60 to 70% of nosocomial infection are associated, in fact, with some type of implanted medical device. Like I said earlier, bacterial biofilm has been observed on most, if not all, such devices, including prosthetic heart valves, orthopedic implants, and cardiac pacemakers. Once biofilm is formed, it's really difficult to treat, and the best solution, most of the time, is just to remove and replace the device. So what's the difference between planktonic bacteria and biofilm? Comparing to a planktonic or solitary form of bacteria, biofilm is a complex three-dimensional structure of a sessile bacteria attached together, making a kind of fortress that protects them for the environment. So how biofilm are formed? There are three main stages for biofilm formation. The stage one, you have a planktonic or free-floating bacteria that will adhere to a biotic or abiotic surfaces. Cell then will aggregate, form what we call a microcolonies, and produce extracellular polymeric substances, also called EPS. And at this moment, the attachment becomes irreversible. During the stage two, a mature biofilm is formed. Cells form what we call a multi-layered clusters. In some cases, we can have a weird form like the mushroom-like form for Pseudomonas aeruginosa biofilm. The last stage is called detachment. It happens when the biofilm reaches a critical mass. So some bacterial cells or pieces of the biofilm itself can be released and be ready to colonize new surfaces and start a new biofilm cycle. Talking about dispersion, during my Ph.D., I worked with Dr. Jacques on Actinobacillus pleuropneumoniae, a big pathogen that produces a remarkable biofilm. Indeed, this pathogen is able to make a mature biofilm in only 4 hours. And more interesting, at 6 hours, this pathogen produces in a higher amount an enzyme called dispersin B that specifically degrades the N-acetyl diglycosamine, also called PGA, which is the main and the most important component of its biofilm, and therefore the biofilm will be detached. Now let's talk about the composition of biofilm. We have, of course, bacterial cells, along with the extrapolymeric substances, or EPS. They are called also the house of biofilm cells because it covers the bacterial cells. So what we can find in the EPS? First, we have water, a lot. In fact, biofilm are highly hydrated, and we can have up to 97% water in biofilm. We also found proteins and extracellular DNA or eDNA. We also have outer membrane vesicles or blebs and ions in bound or free form. All of these provide charges to the structure and could affect the penetration of some antibiotics. Okay, now I will give you some methods to study the biofilms. The most commonly used method to study this form is the static experimental model, also called 96-well plate assay, and the crystal violet staining. So the 96-well plate assay, it's a rapid and highly reproducible system which is extensively used in biofilms. The method is relatively simple. Briefly, from a fresh overnight culture, we usually make a dilution and use this diluted culture to fill the wells of a sterile 96-well plates. Following a static incubation of several hours or days, the culture medium, which contain the non-attached bacteria, it's removed by aspiration, and the plate is washed twice using PBS or water. Then, wells are filled with crystal violet to stain all the attached bacteria. After the removal of the crystal violet solution, the plate is washed twice with PBS or water again and dried at room temperature. To visualize and quantify a biofilm, we add ethanol to the wells and we measure the absorbance of the crystal violet. And here you can see the side view and the upper view of a well containing a biofilm versus a well without any bacteria. You need to know that this system is called a closed system and that after a period of time you will have an accumulation of bacterial products that are toxic for the bacteria. To avoid this problem, dynamic methods or open systems have been developed. The first example is the drip flow reactor. In this method, dilution of bacterial culture are added to channels that contain a glass slide, and cells are grown under a continuous flow of fresh media. This method, this model actually mimics better the in vivo condition, and it's ideal for medical device testing and high biofilm production. Another dynamic system used to study biofilm is called bioflux microfluidics. In this system, the biofilm is under the flow of fresh media, but we are talking about microliter of media this time. And the bioflux is very ideal for live cells analysis, for shear assay, and long-term biofilm cultures. You can see the video on the right where the upper microfluidic chamber contains bacteria grown in a media that actually is not the best condition to promote a biofilm formation. However, in the lower chamber, the media is clearly inducing the biofilm formation. To study the biofilm architecture or its composition, we can also use confocal microscopy. Using different dyes, we can stain bacterial cells or some components of the biofilm, such as the FM143, which stain the bacterial membrane. But we also can use BOBO3 in blue that can stain specifically the extracellular DNA, and the cyperruby in red, which is specifically staining the extracellular proteins. Live dead staining in the middle can also help us to distinguish between the live, the green, and the dead, the red population in the biofilm. And yes, biofilms contains dead cells that are stuck in the structure. In fact, they are considered the main origin of the extracellular DNA and proteins found in the matrix. Another method to study the biofilm architecture is the scanning electron microscopy. I added two images of Helicobacter pylori wild-type strains versus an Helicobacter pylori biofilm mutants, which cannot make biofilm. You can see for the wild-type the three-dimensional structure of biofilm and the way that the bacteria are attached and organized together in this structure. Now let's move to the next section, which is why this sudden interest in this structure. Here you can see this figure that represents the number of publications about biofilm during the last 20 years. And you can see we have a huge increase in the number of biofilm publications in the recent years. So the question is why? To answer this question, I want you to watch this amazing video of Pseudomonas aeruginosa biofilm, and you will probably understand by yourself why this sudden interest. It's amazing, no? These structures, biofilms, are considered by a lot like a city of microbes, a fortress and a shelter that can protect the bacteria from the hostile environment. There are in fact several advantages conferred by biofilms. We can cite genetic exchange, defense against the environment, providing a favorable microniches in a harsh environment like the stomach, for example. But let's focus on antimicrobial resistance and tolerance. First, let's make it clear. Antibiotic tolerance, it's not caused by mutation or inheritance of resistance. It's a transient and non-heritable phenotype defined by the physiological state of the biofilm cells population. In biofilm, in fact, bacteria can tolerate antimicrobials in different ways. First, the biofilm may act as a physical barrier and limit the penetration of some antimicrobials, thanks to the EPS. Bacterial cells in biofilm can also have a slow growth or a dormant state, also called persistent cells, and tolerate some antibiotics that depend on growth rate, such as antibiotics targeting ribosomes or DNA synthesis. On the other hand, we have in biofilm an increase of antibiotic resistance simply because the structure are a reservoir of genetic diversity. In fact, the proximity of cells within a biofilm increase the dissemination of antibiotic resistance genes through the horizontal gene transfers or the integrative conjugative elements. I will now talk about some of my experiments with a biofilm where I found that antibiotics can also be detected as biofilm signals. First, I have to define some terms like MIC. Minimal inhibitory concentration, or MIC, is the lowest concentration of an antibiotic that can inhibit the growth of a bacteria. Below this MIC, antibiotics cannot kill the bacteria, but they can affect the morphology, the surface properties, the pathogenicity, and also the biofilm formation. There are different ways in which a bacteria can be exposed to doses below this MIC, also called sub-MIC. Biofilm, like I said before, can slow the antibiotic penetration, and thus bacteria in the deepest layer of the biofilm may be exposed to sub-MIC. In addition, in some countries, antibiotics are used as growth promoters in animal production for many reasons, such as maintaining the weight gain and feed efficiency, and usually they are given in low doses. During my Ph.D., I worked with Actinobacillus pleuropneumoniae, a pig pathogen, and penicillin G, which is an antibiotic that is used as growth promoter for swine production. And surprisingly, we have found that sub-MIC of penicillin G can increase the production of biofilm by Actinobacillus pleuropneumoniae, a biofilm that actually helps bacteria to survive the antimicrobials and to persist in the lung. I have also found that biofilm may help bacteria to survive the immune response. For this experiment, I exposed macrophages to either biofilm or planktonic population. And the results were really interesting, since we have observed biofilm cells inducing lower expression of pro-inflammatory cytokines. And this suggests that when bacteria makes biofilm, cells in biofilm have better tools to avoid the action of some immune cells. Now let's move to what I'm doing now at UCSC in the Ottemann's lab. I'm working on Helicobacter pylori, and I'm trying to study the biofilm formation and its composition. Let's first talk a little about Helicobacter pylori. Helicobacter pylori has been co-evolving with humans for tens of thousands of years. It adapts itself to survive the harsh environment of the stomach and evade the immune system, allowing it to persist for the life of the host. Even though most of the infections are asymptomatic, Helicobacter pylori persistence is considered as a major risk of gastric and duodenal ulcers and also cancers. Helicobacter infection remain difficult to treat, and when left untreated, they can progress into gastric cancers in 1 to 2% of cases. Helicobacter pylori have also several tools to escape the harsh environment of the stomach, the immune system, and the antibiotic treatments. Biofilm is thought to play a role in Helicobacter pylori pathogenicity, but little is known about this mode of growth. To study biofilm, several researchers have used omics approaches such as metabolomics, proteomics, transcriptomics, and genomics. In our lab, we decided to use transcriptomics to study the biofilm of Helicobacter pylori. For that, we isolated two populations, the planktonic and the biofilm, and extracted their RNA, made libraries, and sequenced them in order to identify signature genes for biofilm formation. By doing that, we have found that biofilm cells are less metabolically active. In fact, genes encoding for the metabolism were downregulated. The biofilm were also under stress and are considered less virulent since virulence genes are also downregulated. And interestingly, we found that genes encoding for flagella were upregulated. That was really weird because flagella, as you know, are used for motility, which is a characteristic of planktonic cells, motile. However, here we have a biofilm, a sessile mode of growth, non-motile. So why flagella in biofilm? To further investigate the flagella's role in biofilm, we use scanning electron microscopy. And you can see here some images of Helicobacter pylori biofilm. And yes, flagella are found in the biofilm, attaching the bacteria or connecting the bacteria together. Because Helicobacter biofilm do not contain polysaccharides, sugar filaments, like other bacteria, maybe they use a flagella instead. To confirm that this is really flagella, we used a fly-M mutant which does not produce flagella. And as you can see, we lost the filaments. And thus, we confirmed that the flagella are part of the biofilm in Helicobacter pylori. We still have a lot of questions, of course, about the role of flagella in Helicobacter biofilm. But for now, we are thinking that flagella, as the images show, could help the bacteria to make a robust biofilm. And that was the fascinating world of biofilm. Thank you for watching.